Vice Agnes, thank you very much for those kind words and the invitation, and to Mihail, who's been very patient as chair of the POC to get me to come to ESPE. He invited me last year, but I'd already got another commitment, and I'm really grateful to him and everybody else for the kind invitation to give a talk here today. The title of my talk is going to be about the calcium sensing receptor, its signaling in physiology and diseases, what I'd like to do before I start telling you about all of this is to just tell you a little bit about the calcium sensing receptor so that we're all, shall I say, working from the same base. So the calcium sensing receptor, it's a G protein coupled receptor, and it's typical of all G protein coupled receptors. It's got an extracellular domain, it's got 7 transmembrane domains, and an intracellular domain. And the extracellular domain in this class of GPCRs is a very large one which forms this bilobed venous flytrap domain, which is where the binding sites for calcium are. Now, the calcium sensing receptor forms dimers, and its main ligand is calcium, but in fact it's not just calcium, it's a lot of cations as well. And in fact, it's a very promiscuous receptor. It binds calcium, magnesium, gadolinium, a lot of other positively charged amino acids, even antibiotics as well, such as gentamicin and things. And you might sort of say, well, how on earth did this arise? Well, it really arose because of its evolutionary origins are that of a nutrient receptor. And that's why it has low affinity for a lot of these ligands there. When it binds to its ligands, e.g. calcium, it signals through a number of G proteins, such as G alpha 11 or GQ or G12, 13, GIO, and GS, and it activates different pathways. And the one I'm showing here, just for simplicity, is activation via PLC beta to activate the IP3 pathway to increase intracellular calcium, and then through mechanisms from there on to alter PTH expression and secretion. And I'd just like you to remember that just for a short while, because this is the assay we'll be using. So when we want to assess the function of this receptor, what we do is we alter the extracellular calcium, and we measure the changes in intracellular calcium. So it's easy. Alter extracellular calcium, measure intracellular calcium, that tells you how that receptor is working. So you can just tuck that away into your brain for a while. We'll come back to that later on. Now, the calcium sensing receptor has a pivotal role in calcium homeostasis, and it is widely expressed, not just in the parathyroids and kidneys, but also the lungs, the pancreas, the heart, the brain, you name it, it's almost found there. So it is widely expressed. And what it does is it has calcitropic roles, i.e. involved in calcium homeostasis, but also non-calcitropic roles, and it's got lungs roles, for example, in lung development, cataract formation, even Alzheimer's, it's being said now, and a number of cancers as well. So what I would like to do is to just focus on the calcitropic roles, and what I want to show is how investigating patients with a variety of calcium-related disorders has taught us about structure-function relationships and insights into these signaling pathways. Now, one of the surprising things about the calcium sensing receptor is that it is really the Henry VIII of the receptors. Now you might sort of say, hang on a minute, he's gone off his rocker, what's he on about? Well, let me tell you this. When I was at school, and when I was a student, we were always taught one gene encodes one protein, and the corollary of that is one disease. And before Henry VIII, the rule always was one king, one queen. This guy went and changed it all. You all know your history and things. He went and had 6 wives, okay, and 6 queens altogether. Well, the calcium sensing receptor is like that. It's not related to one disease. There are many diseases. And just in the calcitropic ones, there are at least 7 disorders, and I can probably tell you about 2 more as well. So these are, for example, familial benign hypocalciuric hypercalcemia, FHH, where you've got a high serum calcium and a low urinary calcium. You can also get adult and neonatal severe forms of primary hyperparathyroidism because of mutations in this receptor. There's another disorder called autosomal dominant hypocalcemia, low serum calcium here, and it may be associated with hypercalciuria as well. A Bartter syndrome, Bartter type 5, and these are the ones that are due to mutations in the calcium sensing receptor. You can also get autoantibodies against the calcium-sensing receptor, and then these can be activating or inactivating autoantibodies, and you can get forms of autoimmune hypocalciuric hypercalcemia or autoimmune hypoparathyroidism. So there are at least 7 phenotypes here with 1 receptor, and I can also tell you some other ones where people have mutations that are completely normal as well. So the spectrum is expanding. So it is really the Henry VIII of receptors. Now, what I'd like to do is to just focus on these calcitropic ones due to the mutations to illustrate a few points. So if you get calcium-sensing receptor mutations, it can give rise to either hypercalcemia or hypocalcemia. And the reason for this is that if you have loss-of-function mutations of the receptor, it causes FHH and neonatal primary hyperparathyroidism. And here's a case of neonatal primary hyperparathyroidism. This is a child at the age of 3 weeks, has got very high calcium, high PTH, and look at these terrible bone erosions that this poor child has got over here. And this needs urgent parathyroidectomy, at least it used to in the old days. Nowadays we can temporize a little bit with a few drugs. But 2 weeks later, at the age of 5-6 weeks, this child underwent a parathyroidectomy, and you can see that 3 months later how well the bones have healed. And this child is now in her 20s and very well and thriving. So this is a very severe disorder of life-threatening hypercalcemia, bone demineralization, and fractures as well. And that's the loss-of-function mutations that can give rise to this. If you get what we call gain-of-function mutations, you get the opposite phenotype. Instead of getting hypercalcemia, you get hypocalcemia, and instead of getting hypocalciuria, you can get hypercalciuria. Now you might as well say, well, gosh, how can this receptor do this? How can it give rise to 2 such diverse phenotypes, one with a high calcium and the others with a low calcium? Well, it depends on what the mutation is doing and where it is. So let me just illustrate this here. I'm going to focus on 2 residues of the calcium-sensing receptor. One is at codon 173, normally it's a leucine, and the other one is at codon 221, it's a proline. There are over 250 mutations reported. I'm not going to go through every one. I just want to focus on these 2 because they illustrate a particular point. Now, if you mutate this leucine at 173 into a proline, you get FHH. If you mutate it instead to a phenylalanine, you get ADH. You get different phenotypes. So here you have the same residue, and you mutate it into 2 different amino acids, and you get completely 2 different phenotypes. And you might as well say, gosh, that's surprising, isn't it? Well, it isn't. You're all pediatricians. I'm sure you still play with Lego sets with your patients, or even at home on your own, but you don't need to admit it right now. But you know when you've got Lego sets, you're playing with those lovely bricks, and you're building a wall. Well, if you're building a wall with those cuboid bricks, and you replace one of those cubes, for example, with a round brick, that's going to give you a weak wall, isn't it? If, on the other hand, you replace it with a hexagonal brick, you'll get as strong a wall, if not even stronger, okay? So proteins are just like building blocks. It depends what you change them to as to whether you're going to strengthen that wall or weaken it. So what's happened here is that when, I told you earlier on, remember, if we, to assess the receptor, we change extracellular calcium, we measure intracellular calcium, which is on here. Here's the wild-type receptor. And we see that what we'll call the EC50 is over here at about 2.5. If you've got a loss-of-function mutation, what this means is it's not that the receptor isn't working, it's working less efficiently. So you need a higher calcium over here to get up to the EC50 over there. So it's working less efficiently. And if you get a gain of function, it's working more efficiently, and you can actually get to the EC50 in a lower way. So the analogy is almost sort of like this, if you want to look at it this way. When I was a younger man, I didn't have much money, and I used to drive a Mini. It was a 950cc engine with gears, and I used to have to get up this hill. I can promise you, no matter how much I speeded up before I got to the hill, by the time I got up to that hill, I had to get down to first gear, okay? Small engine, less efficient, etc., that sort of thing. The Mini is working, but not working very efficiently. Now I'm older and richer, I own a BMW. And I can tell you, I can get up that hill without even changing gear. I'm still in 4th or 5th, okay, yeah? So the BMW is working very efficiently. So Here's my Mini, if you want to look at it that way. Here's my BMW. My gosh, if you can afford it, there's your Lamborghini. All right? Okay, you know. So the receptors are working. It's not that they're not working. It's a question of whether they're working efficiently or less efficiently. So the other thing is this, actually. If we look at so there's clearly, this leucine at 173 clearly has a very important role to play. And the proline, you can get the same sorts of mutations, loss and gain of function here. So what does this tell you about this area? Well, where you're getting these building blocks, it's in the large extracellular domain. You know, I mentioned it was two lobes, lobe 1 and lobe 2, and there's a venous flight trap in between where the calcium came in. Well, these two residues are located on either side of the calcium entry path. So you can see these are gatekeepers. You mutate that into an alternative residue, it can shut the gate and prevent calcium coming in, or you can mutate it and it leaves the gate wide open and it can just flood in. Okay, so that's the, shall I say, the three-dimensional, the structural explanation for it. So by studying patients, we've learned so much about this receptor and also about its structure-function relationships. And of course, this is what we would call the allosteric binding site, and it could be potentially a drug site. But for drugs, we've actually concentrated, because of a number of reasons, on other sites which are called allosteric sites. So allosteric binding occurs at the site of the ligand. Allosteric binding site occurs elsewhere and alters the function of the receptor. And there are two classes of these allosteric modulators. There's one called positive, which is a calcium mimetic, and you already know about this. This is the drug cinacalcet that's already on the market, and it's there to help decrease PTH and plasma calcium. This is the calcium mimetic over here. These bind to transmembrane domains over here, and that's the calcium mimetic decreasing PTH and plasma calcium. You get the other class, which are the negative allosteric modulators called calcilytics, and these increase PTH and plasma calcium. And these are a potential therapy for the hypocalcemia of autosomal dominant hypocalcemia. Because if you treat these patients, they're not hypoparathyroid patients. If you treat them with calcitriol active forms of vitamin D, you will raise their calcium, yes, but you will cause marked hypercalciuria, and that will cause damage to their kidneys. So you've got to be very, very careful how you use that. And we need a better drug than calcitriol for that. So calcilytics offer one such approach. So let me just illustrate how we've approached this. Here, this is a compound made by NPS, 2143, and we have a mouse model for ADH. The mutation is a leucine at 723 over here. And when you express the wild-type receptor, you can see the black line over here, that's a wild-type receptor. And here, the blue line is the mutant receptor, leftward shift, gain of function mutation, a much lower EC50. Well, if we add 20 nanomolar of this drug, fortuitously, we struck it lucky, the curve is more or less superimposed on the wild-type. We've corrected the defect with 20 nanomolar of this drug. We did a dose titration, and by adding 40 and 80, we not only shift this curve to the wild type, but we push it the other way. So here, you get a dose dependency, and if these were patients, these patients would be becoming hypercalcemic. Okay? So this is, in vitro, an effective compound. Well, we all know about drugs. It might work in vitro. Does it work in vivo? Well, we've got a mouse model, so we tested it in that. And here we've given a single dose of this drug to these mice. They're called NUF mice, because they had nuclear flex, they had cataracts. And what we find is that 1 hour after giving the drug, look at this. Wow, the PTH goes up markedly. So here's the untreated heterozygous mice, and these are given drug vehicle only in red, and these are the blue, the ones that are treated. So the PTH goes up in 1 hour, and it comes down. And concomitantly with this, you can see that the calcium also goes up. And I haven't shown the data for simplicity right now, but this plasma calcium is equivalent to that in wild types. So this drug actually works in vivo. And currently, Mike Collins and his group at NIH are undertaking some trials to see its effects in patients. And the most important thing is that not only does it restore normal calcemia, but it doesn't cause hypercalciuria. So this could be a good drug for patients potentially. Now, so much of the calcium sensing receptor. One of the things that happens is that if you've got a patient with FHH or ADH, what you do is you say, oh, well, let's just confirm it, send it off for genetic analysis. And the result comes back sometimes, in 1/3 of cases roughly, no mutation identified. And The simple answer is, to most people say, well, okay, it's not FHH or ADH. No, that's not true, okay? Remember, we are clinicians. We make these diagnoses clinically, and we do tests to confirm it, okay? 65% of FHH patients will have a mutation in the calcium sensing receptor, and about 70% of ADH patients will have a mutation in the calcium sensing receptor. In the other 30 to 35% of patients, there is something else going on. And there is indeed genetic heterogeneity. There are different types of FHH and ADH as well. So FHH is caused by loss of function mutation of the calcium sensing receptor, and this is on chromosome 3q. FHH2 is caused by mutation of G protein alpha 11, and this is on chromosome 19p. And FHH3 is due to adaptor protein 2 sigma subunit mutations, and this is on chromosome 19q. So there's heterogeneity, and there are different types of FHH, and as I'll show you in a moment, there are also different types of ADH as well. So what I want to do for the remaining part of the talk is to tell you about FHH and FHH2 and FHH3. Now, I was very fortunate having some very talented postdocs and clinical training fellows, and that's Andrew Nesbitt, Fadil, and Sarah. We're all working together on this. We took two approaches to try and deal with this. In one approach, we undertook what's called a hypothesis-driven approach, and we identified FHH2 that way, and I'll tell you about that in a minute. And this is the thing that grant-giving bodies like, because they sort of like to have a hypothesis and feel you're smart and all that sort of thing. The reality is, it often doesn't work that way. And what I like to say is, I think equally important are what I call hypothesis-generating approaches, okay? In other words, you don't know where things are, and you've seen some brilliant examples earlier on about exome and next-generation sequencing. And I think these are very powerful approaches. The grant-giving bodies don't like it. They call them fishing trips, you know, but you've just got to persuade them as well. And I'm seeing lots of nods around here that everybody agrees with me. So we used a hypothesis-driven approach and a hypothesis-generating approach to identify FHH2 and FHH3. So let me tell you about that. FHH2 first. So here we took a hypothesis-driven approach. Now, why do we do that? Well, we postulated that mutations of G alpha 11 may cause FHH2, and the reasons were as follows. First of all, the gene causing G alpha 11 is located on the short arm of chromosome 19, which is exactly the location where the disease locus for FHH2 is. So chromosomally, they're in the same region. Of course, it's a big region, and there are about 800 genes there, but never mind about that. At least we were close in that way. But the other thing is this calcium sensing receptor signals through G alpha 11. Wow, well, I mean, you can say, well, that's the next thing in line. Surely that's got to be it. And also, mice harboring paratype-specific G alpha 11 G alpha Q mutations developed hypercalcemia. So we thought this has got to be a strong candidate for this, and we had the family from Salt Lake City and sequenced that, and indeed we found a mutation. Now, this mutation was a 3 base pair deletion. So if you look at the wild type here, you've got AAC encoding for asparagine here. You've got ATC encoding for isoleucine, and an ATC encoding for isoleucine, and then TCC encoding for phenylalanine. That's in the wild type. What happened in the patients is that you had lost one of these ATCs. So this was an in-frame deletion, and you had essentially an in-frame shift over here. So in the mutant, instead of having 2 isoleucines, you had 1 isoleucine and then phenylalanine in the next, So the big question, I'll just take my word for it, this co-segregates with the family. This is the family over here. People who are shaded in have got FHH over here, and these are normals. This is the wild type band, and everybody who's got FHH, for example, this man here and this lady over here, has got a mutant band over here and a wild type band telling you they're heterozygous for this mutation. But the question is this: you lose 1 amino acid. Okay, so you got a protein that's only 1 amino acid shorter. How on earth can this actually give rise to this phenotype? Surely, it can work quite reasonably. Well, we went to do some structural biology analysis, and this is what we found. First of all, this residue here occurs between what's called the beta 2 and the beta 3 sheaths in this loop over here, beta 2, beta 3 loop over here. And when you look at this beta 2, beta 3 loop in evolutionary terms, 2 things happen. Those 2 isoleucines are evolutionarily conserved right the way down to Drosophila and all sorts of things, and also conserved in other paralogs as well. So evolution thinks it's very important to have 2 isoleucines here. Not only that, if you look at this, the length of this loop, it is highly conserved in all species, and it's 13 amino acids long. So what we call this we call this a skipping rope mutation. Now, I'm a great believer in audience participation, so I need a show of hands, please. How many of you have skipped? Oh, you must have skipped, please. Oh, good. Lots of hands going up. Right, okay. So for those people, Muhail didn't put his hand up, so he'll have to explain to him what I'm going to say. I don't think you might know not what a skipping rope is. But look, when you look at a skipping rope, okay, a child is skipping. The length of that skipping rope is very important for that child's height. Gosh, I feel embarrassed telling pediatricians this, but it is. So if it's too short, the child is going to have to skip higher and may not even be able to skip over that. It's going to be inefficient, okay? If the rope is too long, it's going to drag on the ground and be inefficient as well. So, you know, length is important, okay? Biology is just the same. The length of this loop has to be 13 amino acids. And we deleted it here, but I can tell you we've not just deleted this one, we've deleted other ones, we've lengthened it as well. It's got to form this very tight structure with hydrogen bonds that is like this loop, like a skipping rope, and it's got to be exactly that right length for it to function. If you alter that, you mess that around, you get inefficiency. So let me just show you that. Here we've done it in our assay, and I'm just going to show you one. If you're interested in this, look in the New England Journal of Medicine, you see lots of curves when we proved all this. But here's a wild type curve. Nice sigmoidal shape over there, and that's the EC50 here. This is the vector only. And when you do take that mutation, that deletion over here, look, rightward shifter here, loss of function. Okay, so this is G-alpha-11 mutation causing an essential loss of function effect on the calcium sensing receptor, telling you it's not working efficiently. Okay, so that was FHH2. Well, of course, we're not very smart, so we just sort of said, well, are there more? We found more mutations. But the other thing we asked is, well, just like the calcium sensing receptor, when you've got FHH1, loss of function mutations, if you get a gain of function, would you get the opposite phenotype? So we went to our cases with so-called ADH, didn't have a mutation, and cut a long story short, yes, we did. So we found loss of function mutations causing FHH in G-alpha-11, this is FHH2, and we found gain of function mutations as well causing ADH, and this is now called ADH2. Okay? And there have been a number of other papers. You can find these in what we call ADH2, but some clinicians call them hypoparathyroid families, and they've looked at those, and there are a number of those as well that have been identified as well. I would prefer to distinguish them from hypoparathyroidism. So we have FHH1, ADH1 due to calcium sensing receptor mutations, and FHH2 and ADH2 due to G-alpha-11 mutations. So then we went on to our hypothesis generating approach, and here what we did was we mapped, Michael White, a very close friend, had identified a large family in Oklahoma, and my colleagues in Northern Ireland identified another family with FHH, and both these families mapped to chromosome 19q13. So when we mapped the Oklahoma kindred to this region here, it was about 4 million base pairs in size, and you thought, oh wow, this is going to be easy. Well, I always have the unfortunate mishap of landing in regions that are incredibly gene-rich. So there were over 120 genes in this region. When we got the Northern Ireland kindred, we shortened the interval a little bit, but there were still over 110 genes. So what used to happen in the lab was when I got a clinical fellow or a new PhD student, I'd sort of say, look, great project here, 110 genes, you know, have a look at the genes, pick out your 2 or 3 best candidates, identify the gene causing this, and you'll make yourself famous. You know, and that was great, you know, lots of enthusiasm would come in. After about 6 months, sequencing about 2 or 3 genes, that sort of thing, disillusionment would set in, and even with my persuasive powers, I couldn't make them do it. So I'd shift them onto another project that was moving faster. So I calculated, we'd excluded about 10 genes at that stage. I calculated that if I needed to carry on doing this at this rate, I was going to need about 35 to 40 students and fellows, and it's going to take quite a long time. And it's not going to be feasible. Well, fortunately, a new method came along, and you've already heard some excellent examples from the previous 2 speakers about whole exome sequencing. And this is a method, it's brilliant. It enables exons to be captured and enriched from genomic DNA and facilitates high throughput screening. And we did that in this family, and we found a mutation in both these families. So the initial reaction was, oh, it could be a polymorphism. But it turned out not to be. It was a C2G transition in this gene called AP2S1, and it resulted in substitution of a highly conserved arginine residue at codon 15 into a cysteine in this protein called Adaptor protein 2 sigma subunit. So just to show you the co-segregation, we use the restriction enzyme assay for this. The mutant band is up here, and you look at all of these individuals who are affected, and they've got the mutant band, and they've also got all the wild type bands. It's the heterozygous for this. So there's co-segregation of this mutation. When we went on to analyze 50 other FHH patients without CaSR mutations, we found that 11 of those had missense heterozygous mutations, and they all involved this arginine 15 residue. And it changed it to a cysteine, or a histidine, or a leucine. And that was the only things we've ever found in these patients. So this arginine residue is obviously very, very critical. So the question is, what is this adaptor protein sigma subunit, and what is critical about this arginine? Now, one of the things I find about the sort of work that we all undertake in terms of genetics is that one morning you wake up and you're ignorant completely about various things. You suddenly find something like this. You have to hit the papers, read, and the next morning you have to become an expert in it, okay? So if I make some naive statements, do forgive me, but you know, you have to have a very steep learning curve. So what happened here was that this AP2 sigma subunit forms part of a protein called Adaptor protein 2. And this is a heterotetrameric complex composed of alpha, beta, mu, and sigma subunits. And what it does is that after the receptors, not just the calcium sensing receptor, but other ones, have been activated, and you've seen the mutations there, and with the G alpha in them, they signal. And then what happens is that they're internalized for deactivation and recycling. And two proteins are involved. One is beta-arrested, and the other is adaptor protein 2. What adaptor protein 2 sigma subunit does is it binds to dileucine motifs on the cargo proteins. And then it helps to internalize these into clathrin-coated vesicles for further processing. And clathrin-coated vesicles, I mean, to me, they look like club sandwiches, really. You know, the three layers. You've got here the cargo protein and the plasma membrane, so that's the first slice of bread if you want to look at it that way. Here's the adaptor protein over here, the middle layer. And what the adaptor protein does is it's hooked onto the receptor, but it also then hooks onto clathrin over here. So it's pivotal in forming these vesicles for further processing of this receptor. So if this hypothesis was to be correct, that this arginine 15 residue was absolutely critical, then we needed to demonstrate this arginine 15 residue was going to be functionally altering this receptor, and secondly, we need to provide a structural explanation for this. Well, the arginine residues here are absolutely critical in hooking onto these dileucine motifs over here, and if you change it to a cysteine or a leucine or a you actually lose that polar contact, so you will not be able to hook onto that protein and pull it in that way. And what about the functionality of it all? So here we've expressed the wild type and the mutant over here, and I'm just showing you one, but we've done it for all the others. And you see that there's a rightward shift and a higher EC50. So mutation in this arginine residue of sigma subunit here is altering the functioning of the calcium sensing receptor in that way. So that provides us with an explanation of FHH3. Now, one of the other unusual things we found was this, that in FHH3, when we looked at the patients, first of all, they had higher serum concentrations than patients with FHH1. We did this because some of them were actually quite symptomatic from their hypercalcemia. In FHH1, often the patients are asymptomatic, but they were quite symptomatic over here. And the other thing is that in our series, we found a genotype-phenotype correlation. So if the leucine mutation had the highest calciums, then the cysteine and the histidines were amongst the lowest. They're all hypercalcemic, but you can see there's a genotype-phenotype correlation over here. Well, this is all fine and things, but frankly, how does this help the patient? You know? So we thought, well, actually, if they're symptomatic, could we treat them with something? And we decided to use cinacalcet. So we used our assay here, and these are the EC50s, and this is the control, EC50 is here. And these are the mutations in red over here, and when we treat them with cinacalcet, they're shown in blue. You can see the cinacalcet works for every single mutation and restores it to that of wild type. So that's in vitro. So what about in vivo? Well, we had patients with this, and this appeared in the New England Journal of Medicine in April this year, each one with a separate mutation over here. And you can see that, for example, this patient over here with the ARG15 cis mutation, quite severe hypercalcemia, symptomatic, had a parathyroidectomy, didn't really do anything to the calcium, and we treated this patient with cinacalcet, and look, the calcium comes down, and this patient is much, much better. Likewise with this one, a patient here, proband 2, ARG15 histidine mutation, treated with cinacalcet, and right down here. And this patient had the most traumatic of journeys because they had pamidrine infusion, brought the calcium down a bit, but bounced back up again. Parathyroidectomy didn't work, came eventually to the notice of our teams, and treated cinacalcet, and look at that, the calcium's down again. So cinacalcet is very effective at treating these patients. And this is quite unusual because you're talking about a protein that's intracellular. It's not even on the cell surface. It just hooks onto the calcium-sensing receptor, and it's able to modulate the functioning of that complex and to restore normocalcemia. So I hope in the last little while, I've taught you the value of human studies, thinking about them, and dissecting out signaling pathways. And in our case, we've dissected a little bit about the calcium-sensing receptor signaling pathway, and it's told us something about these disorders and the pathophysiology, and that is that FHH and ADH1 are involved in abnormal signaling, and their loss and gain of functions give rise to these disorders of hyper and hypocalcemia. Further down the signaling pathway with G-alpha-11, loss and gain of function mutations give rise to FHH2 and ADH2. And here we're now looking at trafficking mechanisms with adaptor protein 2, where you've got mutations in the sigma subunit giving rise to FHH3. Now, it's been my privilege to tell you about all of this. Of course, the hard work's been done by members of the team and my wonderful collaborators. And I've already mentioned Fadil and Andrew and Sarah, but there's a wonderful team that's working on these projects, and we have lots of visitors who come and join us as well, so please feel free when you want to come and see us. And these are all the wonderful collaborators that we've got internationally who've donated samples, ideas, materials, and science is truly international. And of course, the patients. Thank you very much.